When you think about it, the laws of chemistry and biology and everything else that we know say that it should be gone. It should be degraded completely.
This was powerful evidence that the poster boys for evolution, dinosaurs, hadn't died out millions of years ago, because there's simply no way soft tissue could survive that long. Imagine walking through the Sahara Desert, looking down and seeing an ice cream cone sticking out of the ground. Stunned, you begin to rack your brain for some conceivable explanation as to how it would be possible for it to be sitting here in this environment. Your first thought might be it simply isn't really made of ice cream and just resembles one, but after testing it, you confirm it's truly made of frozen dairy, so you continue to brainstorm. Perhaps there's a camp with a freezer nearby, or maybe it fell out of a low-flying airplane. You might even entertain the idea that someone's developed a drone with an onboard fridge that just deposited this tasty treat seconds before you arrived. The last thing you're likely to do is to question whether you're actually in a desert or not. Now, does all this sound a little funny to you? Well, I can remember just such a scenario playing out, beginning in 2005 when Dr. Mary Schweitzer, a paleontologist from the United States, discovered soft tissue inside the femur of a Tyrannosaurus rex that was supposedly 70 million years old. The inside of the bone wasn't completely filled up with minerals as is usual. And Dr. Schweitzer's team from Montana State University used chemicals to dissolve the bony matrix, revealing the soft tissue still present. Using a pair of tweezers, she demonstrated on camera that the material inside the bone was actually soft and stretchy, completely unfossilized. And then she reported that there were also the remains of blood and blood vessels still intact inside as well. She's been quoted as saying that the blood vessels were flexible and in some instances you could squeeze out their contents. Furthermore, she said the microstructures that look like cells are preserved in every way. Another portion of the tissue was described as flexible and resilient, and when stretched, returns to its original shape. On top of the many dragon legends from people groups worldwide and the copious amounts of petroglyphs, carvings, and other artwork indicating that people have seen dinosaurs throughout history, other than finding a human and dinosaur skeleton buried together, you could hardly think of more conclusive evidence of dinosaurs living recently rather than millions of years ago. Schweitzer's team had presented solid evidence this was indeed soft tissue inside a dinosaur, and a North Carolina State University article confirmed. Colleagues at Harvard successfully sequenced the dinosaur protein that Schweitzer had extracted from the tissue, identifying the amino acids and confirming that the material from the T-Rex was collagen. From a paleo standpoint, sequence data really is the nail in the coffin that confirms the preservation of these tissues, Schweitzer says. This bombshell report ignited a firestorm surrounding the creation-evolution debate, as biblical creationists like myself began eagerly presenting the pictures shown in Science magazine to audiences at our presentations, to help demonstrate that the facts we see in God's world match the history we read in God's Word in Genesis, which includes a young Earth and no mention of the story of evolution. Again, this was powerful evidence that the poster boys for evolution, dinosaurs, hadn't died out millions of years ago, because there's simply no way soft tissue could survive that long, especially in an environment like Hell Creek in Montana where it was found. She admitted as much in a Discovery Channel TV show, discussing what her reaction was upon first observing what was inside the bone.
Blood vessels.
There they are.
Transparent, hollow, pliable, flexible, branching blood vessels that contain small round red microstructures floating in the vessels. I said, this is not possible. Do it again. When you think about it, the laws of chemistry and biology and everything else that we know say that it should be gone. It should be degraded completely.
Now, unsurprisingly, some evolutionists downright denied what Schweitzer said she discovered. So unlikely was her claim given the evolutionary timescale. I had one reviewer tell me that he didn't care what the data said. He knew that what I was finding wasn't possible, says Schweitzer. I wrote back and said, well, what data would convince you? And he said, none. And behind this denial came the first proposal from Bible skeptics of why it mustn't be what Schweitzer's research showed it as, that what she'd found must be the result of some type of biofilm, simply some type of intrusive organic material that had made its way inside the petrified remains of the T-Rex. This dismissive explanation quickly got out into the atheistic community as a foil against those misleading creationists, despite her findings being in one of the most distinguished evolutionary journals on the planet, with peer-reviewed research behind her, no less. I can remember having several skeptics shout it out when I was doing presentations. That's just biofilm. It's not dinosaur soft tissue, was the challenge, along with the usual accusations of false reporting, despite the fact I was quoting directly from Schweitzer's article, she herself being an evolutionist, and showing the pictures published in it in their full and complete context. This quickly raced around the internet as well and was the go-to comeback for evolutionists wanting to rain on the creationist parade of this mind-blowing evidence that was in complete contradiction to the evolutionary story. However, shortly thereafter, the biofilm brigade got permanently silenced as Schweitzer's team made an even more staggering discovery in 2009. This time, they found soft tissue, blood, and proteins inside of a supposedly 80 million year old hadrosaur, a duck-billed dinosaur. And to quell all doubts, due to the aggressive pushback she'd received from her fellow evolutionists the first time, despite her reputation as a careful and experienced scientist, Schweitzer's team was meticulous in their work, making sure to account for anything that could discredit her research. The chief of the Division of Matrix Biology at BIDMC, an independent lab used to verify the find, reported, "Because I'm a collagen biochemist, our lab was contacted to perform an independent analysis of this new bone find. We isolated the proteins, collagen, laminin, and elastin from the bone and also extracted bone cells and blood vessels from this sample. Our findings demonstrated that it did contain basement membrane matrix." What Schweitzer's team had demonstrated was above reproach. Her former evolutionary detractors' minds were changed, and the biofilm boys quickly tucked their tails and ran, never to be seen at a creationist presentation again, despite the bravado they'd previously shown. And after this, the dam broke wide open. Just a year later, National Geographic had an article titled, "Many Dino Fossils Could Have Soft Tissue Inside." And by 2015, evolutionists were reporting on how an accidental discovery of blood and collagen in several dinosaur bones could rewrite textbooks. They further said, "It suggests that nearly every fossil science studied in the past century could contain similarly well-preserved blood and tissue samples." All this to say, soft tissues have been found in dinosaurs, or dinosaur era, according to the evolutionary timeline, creatures now well over 100 times. These have been found in many different types of organisms, ranging from dinosaurs to mammals, birds, plants, reptiles, amphibians, clams, insects, and arthropods, sponges, and worm fossils. Not only that, but they've also been found all over the world, from China to Mongolia to Russia, Madagascar, Europe, the UK, and all over North and South America. Which means they've been exposed to a wide variety of environmental conditions: cold, hot, wet, and dry. And even further, they've been found throughout the entire fossil record, from the Cambrian to the Triassic, Jurassic, and Cretaceous periods. And they've also been found at different levels, the oldest being a marine worm claimed to be 551 million years old. All of this is totally devastating to the entire evolutionary narrative, because according to a 2002 report in the science journal The Biochemist, even if collagen were stored at 0 degrees Celsius, it would not be expected to last even 3 million years, let alone 65 million, and certainly not over 500 million like our wormy friend is supposed to be. In a more recent 2011 article, it's been experimentally established that at 10 degrees Celsius, around 50 degrees Fahrenheit, only 1% of the original collagen in a bone sample can survive for longer than 700,000 years in an optimal burial environment. But as mentioned, it's a tad hotter than that in Hell Creek, where Schweitzer's soft tissue was recovered. These facts have been a thorn in the evolutionary community's side for several years now, as they're incredibly difficult to explain within an evolutionary millions of years time frame. Dinosaurs are said to have gone extinct 65 million years ago. Dinosaurs are said to have evolved into birds. Dinosaurs are an evolutionary icon, and their fossils are the stuff of legend in the atheistic humanistic worldview that teaches materialism is absolute. So the discovery of soft tissue inside them is a kill shot for the idea of deep time, which is a requisite for the story of evolution. Because everyone knows soft tissue breaks down very rapidly, certainly much faster than millions of years. But without millions of years, evolution would be impossible. So what to do then for the naturalist? Evolutionists recognize the soft tissue issue is a serious threat to their materialistic worldview because it defies known experimental decay rates, and they know that biblical creationists have been using it effectively to demonstrate the falsehood of the story of evolution. As one science journal noted, despite the consistency of these data, the endogeneity, the dinosaur native proteins, of these sequences has been questioned and remains controversial. This is in part because of the ages of the specimens, which are well beyond the proposed theoretical limits for protein survival in bone. So they're fighting back by claiming they've discovered several ways these soft tissues could have been preserved, with one article telling educators to explain them to students so they won't be influenced against evolution. These protective factors can result in soft tissue preservation that lasts millions of years. It would benefit educators to be aware of these phenomena in order to better advise students whose acceptance of biological evolution has been challenged by young Earth creationists' arguments that are based on soft tissues in dinosaur fossils. Indeed, there's been a spat of popular-level articles claiming that Dr. Schweitzer and others may have found the answer to the soft tissue issue. For example, Schweitzer has proposed that iron might help preserve dinosaur soft tissue, both by helping to cross-link and stabilize the proteins, as well as acting as an antioxidant. She's tested this idea and published articles to summarize her new hypothesis. Schweitzer's latest research shows that the presence of hemoglobin, the iron-containing molecule that transports oxygen in red blood cells, may be the key to both preserving and concealing original ancient proteins within fossils. Another article explained further. The free radicals cause proteins and cell membranes to tie in knots, Schweitzer said. They basically act like formaldehyde. Now, formaldehyde, of course, preserves tissue. It works by linking up or cross-linking the amino acids that make up proteins, which makes these proteins more resistant to decay. In a technical paper, Schweitzer claimed that hemoglobin increased tissue stability more than 200-fold, from approximately 3 days to more than 2 years at room temperature. However, even under moderate scrutiny, Schweitzer's explanation quickly falls to pieces. In her new paper, she discusses experiments that appear totally unrepresentative of the conditions under which these dinosaur remains were actually preserved. Here's a description of what her team did. They soaked one group of blood vessels in iron-rich liquid made of red blood cells and another group in water. The blood vessels left in water turned into a disgusting mess within days. The blood vessels soaked in red blood cells remained recognizable after sitting at room temperature for 2 years. But from reading the supplementary material in her article, it appears that pure hemoglobin was used, not lysed cells or material that could be expected to mimic what would be present in an animal carcass. Blood vessels soaked in laboratory-prepared hemoglobin is hardly representative of decomposing bones. One might, or should, also ask, how realistic a concentrated hemoglobin extract is compared to what happens inside dead bodies we see in the real world. While unrealistically concentrated hemoglobin might preserve for a time, it doesn't follow that natural dilute hemoglobin will act the same way. Coroners, for example, observe that tissues rich in blood vessels such as lungs and gills often decay very quickly. And the suggestion that blood vessels remaining recognizable for 2 years somehow demonstrates that these could last 35 million times as long requires a phenomenal cognitive leap, let alone a full 225 million times longer, which is what our amazingly decomposition-resistant marine worm would have required. Furthermore, it's not plausible that iron could be as good a preservative as formaldehyde, which directly forms covalent crosslinks between protein chains, something iron can't do. But even if we granted it had the same preservative power, just for the sake of the discussion, what reason is there for anyone to expect that formaldehyde could preserve soft tissues for millions of years? Embalmers of human bodies widely acknowledge that their use of formaldehyde is to slow down, not prevent, the relentless process of decomposition. And what's our best example of just how well that works? Well, in a Scientific American article discussing the preservation of the not yet 100-year-old body of Vladimir Lenin, the communist revolutionary and founder of the Soviet Union, they mentioned the following. A core group of 5 to 6 anatomists, biochemists, and surgeons, known as the Mausoleum Group, have primary responsibility for maintaining Lenin's remains. The Russian methods focus on preserving the body's physical form, its look, shape, weight, color, limb flexibility, and suppleness, but not necessarily its original biological matter. The body gets re-embalmed once every other year, a process that involves submerging the body in separate solutions. Each session takes about 1.5 months. So, if it takes a team of dedicated experts to constantly try to slow the degradation of Lenin's corpse, it's obvious that chemical decomposition will relentlessly occur in soft tissues, regardless of whatever special factors, natural or man-made, might be helping to preserve them. Over millions of years, the lack of enzymatic and bacterial degradation makes no difference. DNA and proteins will eventually succumb to ordinary chemistry, especially reactions with water, which several of the soft tissue finds were undoubtedly exposed to. And evolutionists have likewise admitted this. After cell death, enzymes start to break down the bonds between the nucleotides that form the backbone of DNA, and microorganisms speed the decay. In the long run, however, reactions with water are thought to be responsible for most bond degradation. Groundwater is almost ubiquitous, so DNA in buried bone samples should, in theory, degrade at a set rate. So Schweitzer's mechanism holds that iron atoms help cross-link biochemicals, including proteins, making them larger, tougher molecules. But there's also a newer idea that holds that the same kind of reactions that transform sugars and lipids into bread crust in an oven also occurred in some fossils, and so may account for this miraculous preservation of soft tissues for up to 500 million years. These, what are known as Maillard reactions, happen when proteins react with sugars to form dark-colored polymers. It's what puts the outer crust on French fries, makes caramel, and creates bread crusts. Hence its moniker, the toast model. However, neither the iron nor the toast model directly preserves the original biochemistry. They instead transform it into biomaterials that would preserve merely the original shape, like formaldehyde and other solutions due to Lenin's body, for example. So both mechanisms don't actually account for much of what they're actually finding inside these supposedly millions of years old creatures. This original soft tissue that hasn't been transformed into tougher biomaterial. So what's their solution? Well, some now propose that perhaps these two models somehow work together to create some kind of shielding effect that preserved the original non-cross-linked, non-toasted soft tissue. However, the Maillard effect happens between 280 and 330 degrees Fahrenheit, which, granted, are temperatures that some fossils may have reached while buried deep beneath the earth. But what happens if you don't take the bread out of the oven in time, so to speak? Every baker works with a timer of some kind, and on average it takes less than 35 minutes to bake a loaf of bread and get that nice crusty outer layer. But what if we left it in the oven on and off for 65 million years? And what would happen to a pot roast, analogously the iron-soaked material, we left in there with the bread? Even a roast that had been soaked in formaldehyde. Remember that Schweitzer's iron-soaked material was left out at room temperature. And what about the soft tissues still there that aren't iron-soaked or toasted? Are you seeing the utter ridiculousness of these scientific proposals about what could somehow preserve these soft tissues? To the general public, the power in this argument is its seeming simplicity. The average Joe might think, oh, I get it, iron acts as a preserving agent like formaldehyde, the stuff scientists use to embalm things. It's like those animals preserved in jars I've seen in laboratories. So the iron in the dinosaur's blood must have preserved the organic material, and scientists know what they're talking about more than I do, so dinosaur soft tissue makes sense to me. It's strategic. Just like the evolutionists I quoted earlier, who warned educators to let students know how soft tissue preservation happened, despite not having a lick of long-term observational experimentation, by announcing, "We already have the answer." This is how evolutionists tried to lessen the impact of the argument. From now on, Joe will likely not be surprised if he's presented with the facts of dinosaur soft tissue found in fossils, thinking evolutionary scientists have already explained this, even if they don't have a realistic understanding of how tenuous their proposals are. The takeaway is that, obviously, the creationists are crazy to think that dinosaurs died out recently because, science. However, think about it. Take a laboratory-prepared specimen, place it in a jar full of formaldehyde, then stick it in the ground encased in rock. And just for good measure, keep the surrounds permanently frozen at 0 degrees Celsius. It would still be subject to thermodynamic breakdown of such complex, fragile molecules. Atoms and molecules in a compound are always in motion, even at such freezing temperatures. For any scientist to have said prior to Schweitzer's discoveries that they would have expected blood vessels, delicate cell structures, and proteins after 70 million years from such an experiment would have been inviting derision at best and psychiatric scrutiny at worst. There are very good scientific reasons behind Schweitzer's earlier comment. When you think about it, the laws of chemistry and biology and everything else that we know say that it should be gone. It should be degraded completely. So what has Dr. Schweitzer and others shown with her more recent iron observations? Well, she's demonstrated that the iron in red blood cells apparently has some qualities that could contribute to soft tissue preservation, at least if it's artificially concentrated. And there may be legitimacy in some of the other models proposed as well. But far from being a threat to biblical creationists, this may well be a plus. Because this type of research may eventually help explain how such fragile things could possibly last the approximately 4,400 years since Noah's flood. Because seeing proteins and even cell microstructures after so long is still surprising. But again, it isn't as surprising as believing they could have lasted over 500 million years. Perhaps the most important lesson in all of this is the power of the paradigm. Metaphorically, evolutionists find themselves strolling through a desert surrounded by freezing cold ice cream cones popping up everywhere as far as the eye can see. But trying to get them to admit they aren't actually in a desert goes against everything their worldview is based on, i.e., the ideology of millions of years. Indeed, in the face of today's widespread secular religion, admitting that the Earth isn't very old would have the ideological impact of a nuclear warhead. The story of evolution desperately needs millions of years. And in the face of this evidence, they continue to clutch at straws to explain it, no matter how ridiculous the proposals are. Needless to say, the soft tissue found all over the world in fossils at all levels fit beautifully within a biblical young Earth timescale, because the facts fit perfectly with a worldwide event that essentially, simultaneously within a year, buried billions of creatures together rapidly and completely just a short while ago. These soft tissue finds are the remains of creatures that were buried during the Genesis flood, approximately 4,400 years ago, showing once again that what we see in God's world matches what we see in His Word.